Hi class, and welcome to Chapter 8, which will be on the cellular basis of reproduction and inheritance. We'll be covering a couple of things that we talked about in Lab 9, cellular respiration. So we're first going to kind of introduce this chapter, we'll talk about cancer cells. They start out as normal cells, but different mutations in genetic material can cause them to lose the ability to regulate their division. Cancer cells divide and may spread, invading other tissues, disrupting organ function, and eventually killing the host. And although uncontrolled cell division is harmful, normal cell division is necessary in all forms of life. And this is a look at some cancer cells and asking ourselves, can cancer therapy be personalized for a specific genetic code that someone might have for cancer? So in this chapter, we'll be going through cell division and reproduction, the eukaryotic cell cycle and mitosis, meiosis and crossing over, and alterations of chromosome number and structure. And this PowerPoint is a little long, so we might do it into two parts here. So first, talking about cell division and reproduction. Cell division is at the heart of the reproduction of all cells and organisms because cells originate only from pre-existing cells. Some organisms reproduce through asexual reproduction, which means their offspring will all be genetic copies of the parent and identical to each other, or clones. And other organisms reproduce through sexual reproduction, creating a variety of unique offspring. So a yeast cell producing a genetically identical daughter by asexual reproduction is shown here. Here is a look at a sea star reproducing asexually via fragmentation and regeneration of a body part from the fragmented arm. And here's a look at an African violet plant reproducing asexually after it's been cut a little bit and it'll regrow. Sexual reproduction produces offspring with unique combinations of genes, as shown in this picture here. And here's a look at the dividing cells in an early human embryo, which is kind of a cool picture to see. Here's a human kidney cell dividing. Prokaryotic cells reproduce asexually by what we call binary fission, which is a term that means dividing in half. And in typical prokaryotes, most genes are carried on one circular DNA molecule that, with associated proteins, constitutes the organism's chromosome. And as the cell replicates its single chromosome, the copies will move apart, the plasma membrane pinches inward, and more cell wall is made, which eventually divides the parent cell into two daughter cells. And binary fission is classified as asexual reproduction because it'll make a completely identical offspring. So here's just a look at binary fission of a prokaryotic cell, step 1, continued elongation of the cell, movement of the copies, and eventually division into two daughter cells. So this is binary fission. And this is a look at electron micrograph of a bacterium in a late stage of dividing. Then the eukaryotic cell cycle and mitosis is a little more complex. A eukaryotic cell has many more genes than a prokaryotic cell, and they're grouped into multiple chromosomes in the nucleus. Each eukaryotic chromosome contains one long DNA molecule, and individual chromosomes can be visible under a light microscope when the cell's in the process of dividing, but otherwise these chromosomes are normally very thin and loosely packed into what we call chromatin fibers that are too small to be seen. Before a cell does start dividing, the chromosomes will duplicate, producing sister chromatids, which contain identical DNA pieces that are joined together along their lengths. And cell division involves the separation of sister chromatids and always results in two daughter cells, each containing a complete and identical set of chromosomes. So here's a look at a plant cell from a lily just before cell division, and you can see the condensed chromatin, which contains all the genetic material. Here's the look at chromosome duplication and distribution. So the sister chromatids are attached together at the centromere. The chromosome is 1, and separation of sister chromatids into two chromosomes distributes them into two daughter cells. Here's another look again about how the chromosomes form sister chromatids, and then they separate into two chromosomes to distribute into two daughter cells. Here's a transmission electron micrograph of the sister chromatids. The cell cycle is an ordered sequence of events that run from the time a cell is first formed from a dividing parent cell into its own division into two cells. And we'll go through the cell cycle now. So here's the eukaryotic cell cycle. The majority of the time, the most cells spend in what we call interphase, the G1S and G2 phase. And then the mitotic phase is actually the phase when the cell will divide into two. Mitosis distributes duplicated chromosomes into two daughter nuclei. And after the chromosomes are coiled up, a mitotic spindle made of microtubules moves into chromosomes to the middle of the cell. And the sister chromatids then separate and move to opposite poles of the cell, at which two new nuclei form. So here's a look at the stages of cell division by mitosis. The interphase is the normal, kind of what the cell does is its normal phase. And then the mitosis phase, we're going to start to get through. The first phase is prophase, and then the next phase is pro-metaphase. Here are the stages of cell division, interphase through pro-metaphase. The 6 stages are as follows: interphase, mitosis, prophase, and then going through the phases. But the first couple stages in interphase are that we have the chromatin condensing into chromosomes, and we have the mitotic spindle about to form. The mitotic spindle gets into place as the chromosomes then start to move toward the center of the cell. And here's a look at the interphase stage. The prophase stage, so we start to see the chromosomes condensing in the middle of the nucleus, as shown here. They kind of look like little worms. Here's pro-metaphase, they're condensing even more, and we see the mitotic spindle starting to form. And that mitotic spindle is what will split the cell apart. So here we have the next couple stages of mitosis. Metaphase is when the chromosomes line up on what we call the equatorial or metaphase plate. Anaphase, the chromosomes will be split or pulled apart. And then telophase and cytokinesis are when two new daughter cells will form. And that is shown by these. So here's a look at the metaphase stage. The chromosomes are lining up across the middle of the cell. And then anaphase, you can see the chromosomes being pulled apart to either side. And here we have telophase and cytokinesis. We start to see the formation of a new cell wall here to eventually form two daughter cells. And this is a great animation of mitosis that it's fun to see. I'd encourage you guys to watch that. Here's a look at a sea urchin, a time lapse. So cytokinesis differs for plant and animal cells. And cytokinesis literally means the splitting of the cell into two. Cytokinesis overlaps the end of mitosis. In animals, cytokinesis occurs when a cell constricts, forming a cleavage furrow, and in plants, a membranous cell plate forms and then splits the cell into two. So here we look at the cleavage of an animal cell in cytokinesis, forming two daughter cells. And you can see the cleavage furrow. That just means the part which will form a new cell wall to separate these two cells into two. And here's a look at the cleavage of the animal cell. And in comparison, the cell plate formation of a plant cell. So in a plant cell, a new cell plate forms to form a new cell wall. So it forms a kind of a new cell wall from within. Whereas in an animal cell, we kind of pinch off the cells to form two different cells. So that's kind of the big difference there. So the cell plate formation in a plant cell. And here's a look at cytokinesis, which will compare the two, a plant and an animal cell. In laboratory cultures, most normal cells divide only when they're attached to a surface. So the cultured cells will continue dividing until they touch one another. Most animal cells divide only when stimulated by growth factors, and some do not divide at all. The growth factors will stimulate the cells to divide. And I skip over some of those checkpoint questions as they're not always relevant. You can go back and look through them if you would want to. Here's just a look at an experiment demonstrating density-dependent inhibition, which just means using animal cells grown in a culture. Removing of some of the cells will start to restore the next layer of cells. And it's kind of neat to see this because then you can see how cancer cells can form and they will clump together, overlapping other cells. Here's a look at another experiment demonstrating the effect of growth factors on the division of cultured animal cells. A growth factor will help the cells to divide even more quickly. And you can see that here the cells are failing to divide in this side of the experiment, but with the addition of the growth factor, it's helping those cells to divide. So growth factors, what are they? They will signal the cell cycle to continue or to stop. So they're kind of the control system. The growth factors are a set of proteins within the cell that controls the cell cycle. They will signal, their signaling affects critical checkpoints in the cell cycle, which will determine whether a cell will go through the complete cycle and divide. And the binding of growth factors to specific receptors on the plasma membrane is always usually necessary for cell division. So here's a look at a schematic model for the cell control growth cycle. We have these checkpoints. And those checkpoints will be these growth factors, which will continue to control the cell to go into a further kind of another part of the interphase or mitosis. So here's a look at how a growth factor signals the cell cycle control system. As a receptor protein is embedded in the plasma membrane, the growth factor binds to that receptor protein, which sends relay proteins and signals a transduction pathway to trigger the control system of the cell. Cancer cells will divide excessively to form masses called tumors. Malignant tumors, which are cancerous, can divide and invade other tissues, and radiation and chemotherapy are very effective as cancer treatments because they interfere and try to stop the cell division of those cancer cells. So here's a look at the growth and metastasis or spreading of a malignant tumor of the breast. So here's the tumor and how it can invade surrounding tissue of the breast, and then how eventually it can metastasize, enter the lymph structures, and the tumor can be found in another part of the body. Mortality rates from cancer vary by the age of diagnosis, your race, and other factors, and taking such data into account may improve outcomes of cancer treatment. Here's a look at the risk of death from breast cancer 20 years after the diagnosis, and breast cancer death rates for subsets of DSIS patients. So it's taking a look at the age at the diagnosis and your 20-year death rate or survival rate, and then the ethnicity. So what this is saying is just getting early detection is really important. Meiosis then and crossing over. So meiosis has to do with cellular reproduction in the gametes or the sex cells, your sperms and eggs. And the somatic cells of each species contain a specific number of chromosomes. For example, human cells have 46, consisting of 23 pairs of homologous chromosomes, and the chromosomes of a homologous pair of autosomes carry genes for the same characteristics at the same place or locus. So here's a look at a pair of homologous duplicated chromosomes, how a locus contains the same pair of genes at the same point on the chromosome. Cells with two sets of homologous chromosomes are called diploid, and gametes, or your eggs and sperm, are haploid cells with a single set of chromosomes, and the sexual life cycles involve the alteration of haploid and diploid stages. So here we look at meiosis forming different egg and sperm cells. And then at first, so these are haploid, meaning they contain half the number of chromosomes. And then at fertilization, an egg and a sperm come together to create the diploid zygote, or the total number of chromosomes or sets of chromosomes needed. So here's a look at how meiosis halves chromosome number through two sequential divisions. So what meiosis does is it takes a parent cell and it divides it twice, so that we start with a kind of a full set of chromosomes, and then the 4 haploid cells are the daughter cells, which only have half the set of chromosomes. Meiosis, like mitosis, is preceded by chromosome duplication, but in meiosis, the cell divides twice to form 4 daughter cells. The first division starts with the pairing of homologous chromosomes, and in crossing over, homologous chromosomes exchange corresponding segments. So meiosis 1 separates the members of each homologous pair and produces two daughter cells, each with one set of chromosomes. And meiosis 2 is essentially the same, whereas in each of the cells, the sister chromatids of each chromosome separate, and the result is a total of 4 haploid cells. So here we just have a look at the stages of meiosis part 1, taking us through interphase, and then meiosis 1, which is where the homologous chromosomes will separate. And this will take us through the stages of meiosis one at a time: prophase 1, metaphase 1, anaphase 1, and then we'll go into telophase 1 and cytokinesis, and then we'll go through meiosis 2, which is where the sister chromatids separate. So we'll go through telophase 1 and cytokinesis, and then meiosis 2 is where we will separate the sister chromatids. Here's two lily cells undergoing meiosis 2. So you can see there are two sets of chromosomes separating. Both mitosis and meiosis begin with diploid parent cells that have chromosomes duplicated during previous interphase. So mitosis produces two genetically identical diploid somatic daughter cells, and meiosis produces 4 genetically unique haploid gametes. And this, I think, we're eventually going to fill in this chart that take us through a parent cell and what happens as it goes through mitosis or meiosis to compare the difference between the two. And this is a great chart to study to compare and contrast the two different types of cellular division, whether it's mitosis or meiosis. And again, it's just a good thing to remember is that mitosis is what any cell in your body goes through, and meiosis is what your gamete cells go through, your sex and your sex cells, whether that's your sperm or your eggs. Similarities and differences between the two. Another good chart to focus on. Each chromosome of a homologous pair differs at many points from the other member of the pair. Random arrangements of chromosome pairs at metaphase 1 of meiosis lead to many different combinations of chromosomes in eggs and sperm, and random fertilization of eggs by sperm greatly increases this variation. So here's a look at the results of the independent orientation of chromosomes at metaphase 1, at metaphase 2, and then the end results. Here's a great video on genetic variation to kind of help with that as well. The differences between homologous chromosomes come from the fact that they can be different versions of genes at corresponding loci or places. And crossing over is what we call an exchange of corresponding segments between non-sister chromatids of homologous chromosomes. And this is what I mean by that. So right here, we have a tetrad in parent cell. And we'll see the coat color of genes is either brown or black. And then the parents will create offspring that will either be brown coat with black eyes, white coat with pink eyes. So here's the differing genetic information, the coat color and eye color, and a tetrad of chromosomes in a parent cell is a homologous pair of duplicated chromosomes. One chromosome codes for a brown coat color, C, and a black eye color, big E, and the other chromosome codes for a white coat color, small c, and a pink color, little e. And after meiosis, each daughter cell is separated, and two sister chromatids will give the offspring a brown coat and black eyes, and the other two chromatids will give the offspring a white coat and pink eyes. So here's differing genetic information on homologous chromosomes producing two different types of offspring. This shows the sites of crossing over, and that's called the chiasma, the place where the chromosomes cross over. And a tetrad of chromosomes makes sister chromatids into chromatids. So you can see this crossing over is shown here, the chiasma. All right, so here's a look at the crossing over and just explaining how it increases genetic variability even further. And that's because the chromosomes are crossing over and they are switching out genetic information from each other. And this is a great animation on crossing over. More than anything, this chapter will probably be really helpful to download and watch all the animations. Alterations of chromosome number and structure. An abnormal chromosome count is the result of non-disjunction, which can result from the failure of a pair of homologous chromosomes to separate during meiosis 1, or the failure of sister chromatids to separate during meiosis 2. And this is a look at non-disjunction, when the failure of the chromosomes, they fail to separate. To prepare a karyotype, which is a photographic inventory of an individual's chromosomes, white blood cells will be isolated, stimulated to grow, and then stopped at metaphase 1 or metaphase and photographed under a microscope. And the chromosomes are arranged into ordered pairs so that any chromosomal abnormality can be detected. So here's just a look at how we prepare a karyotype, which will show us a person's kind of picture of all their chromosomes. And this can be really important for seeing if any genetic abnormalities will occur in the person, because you can take a look at their chromosomes and see if anything is missing. For example, trisomy 21 is the most common chromosome number abnormality, and this results in a condition called Down syndrome. And a karyotype showing trisomy 21 is shown at the top, where we kind of have on chromosome 21 an extra chromosome and an individual with Down syndrome. So here we have a zoomed in look at the chromosome number 21, and we have an extra chromosome within that. So trisomy just means 3. Here's the maternal age and incidence of Down syndrome, just showing many times, not always, a mother's age can play a role in affecting if the child will be born with Down syndrome. Non-disjunction of the sex chromosomes during meiosis can result in individuals with a missing or extra X or Y chromosome. And in some cases, such as XXY, this leads to syndromes that can affect the health of the individual. And in other cases, such as XXX, the body can be normal. So here's a look at sex chromosomes, abnormalities, the number in humans, the syndrome, the origin of non-disjunction, and then the symptoms. So these are some abnormalities of the sex chromosomes. Non-disjunction can also produce polyploid organisms, which are organisms with an extra set of chromosomes. And such errors in cell division can be important in the evolution of new species. Here's a gray tree frog, a tetraploid organism. Chromosome breakage can lead to other rearrangements and translocations that can produce genetic disorders, cancer. And here's just a look at some of the alterations of chromosome structure and how they can lead to some problems later on. Deletion, deleting out a segment of the gene, duplication, inversion, switching it out the orientation, reciprocal translocation, the translocation associated with chronic myelogenous leukemia, for example, which is a cancer. And after this, there's a lot that we went through in this chapter. And again, this at the end of the chapter gives you a good kind of review of what you should know, as well as some career options that you could go into if this is interesting to you, as well as some kind of a review of some of the figures we went over, some good connecting concepts, questions, and some good graphs to kind of take you through some of the concepts that we went over as well. So thanks for listening, and I hope you guys are all doing well.